I tend to digress. We're going to make 1,000 copies of how to react to the flu virus. So what's the next step? We need to send these Xerox copies from the nucleus, or the safe where we keep our blueprint. We need to send it outside to the cytoplasm where the factories are located. Basically, the factories are going to they're going to help the body to react to the flu virus. In this case, what they're going to do is they're going to make chemokines. Now again, this is another topic, or this is another point that's going to be a repeating theme in this class. Chemokines. And the two important ones that we have are the CC chemokine and the CXC chemokine. I highly recommend that you keep these straight. This is something that you can easily confuse. So try and keep the CC and the CXC chemokine and their respective receptors on the macrophage and the T helper cells correct. And we're going to talk more about that here in a moment. But as a hint for the test, try and focus in on these. Okay, so the nose cell sends out these chemokines, and the CC chemokine is recognized by the CCR5 satellite on the macrophage. And then the CXC chemokine is recognized by the CCR4 satellite on the T helper lymphocyte. I'm sorry, the CC chemokine is also recognized on the CCR5 satellite on the T helper lymphocyte. And this is really important to know that macrophage is only CCR5, something you're going to want to study, and that T helper lymphocytes have CXCR4 and CCR5. So you're going to want to keep these straight. This is a repeating theme in this class, and we're going to get more, when we start talking more about the biology of HIV, these are going to become particularly important. Okay, so, and I'm sorry, these, the macrophage and the T helper lymphocyte, we're going to talk more about these, but these are white blood cells. These are the immune cells that the body needs in order to fight infections. So, when they receive these chemokines, it basically tells them that the body has an infection and that they need to react and fight they need to be activated and start, they need to get involved in fighting the infection. What they do is they basically make track shoes. What are those called? Pseudopods? Yeah. Something like that. So they start making track shoes, which and you know, here, like Dr. Jacobs said, there's just two, but really there's going to be thousands, maybe millions, that will help carry these cells to the site of infection. And these track shoes are not activated, these track shoes are activated only when they receive the chemokine receptor. And I guess also chemokine is another vocabulary word that you're going to want to keep straight. The chemokine is made by the cell. The chemokine is not a lymphokine. It is not a lymphokine. This is a chemokine. Okay, so they're activated by chemokines. They make track shoes, which carry them to the site of infection. And what more did we? Okay.
All right. So, is it captured properly? Mm-hmm. Okay. So we'll pick up on the immunology lecture from January 13th. And it picks up right where we left off, talking about cells racing to the site of infection. So if we have an infected area, say we use the example of a virus infecting an epithelial cell in the nose, cells would be told to make track shoes and race to the site of infection through chemokines that we talked about. So chemokines, again, we'll recap and say that those are signals that the cell is sending out once it gets infected. So you can think of it almost as a cellular screen, that the cell is saying, here, I'm infected, come and take care of whatever the foreign antigen that has done the infecting, come and take care of that. So we'll talk about innate immunity first. And it's real important to keep in mind, as we apply this to HIV, the concepts of innate immunity and active cellular immunity. So in the back of your mind as we're going through this, please differentiate between the two and we're going to talk about HIV really affecting our abilities as cellular immune systems go to mediate disease, whereas innate immunity, as we'll see, is not going to be as affected. So
it affects what were we calling that again? Not the innate immunity, but the
specific
immunity. Okay.
So we can say specific immunity is like that cellular mediated immunity.
Okay.
So we'll use those words in turn. So the primary cell that gets things kicked off, that gets the party started, is the macrophage. So the macrophage is not going to be specific. When it's activated and it rushes to a site of infection, it's going to chomp up everything in its path and try to sample pieces of the pathogen. So it's going to be eating up cells, like our cells. But in some of those cells, there will be viruses. While we're looking at the macrophage, I'm also might say macrophage, either is correct. CD4 and CCR5 are present. And again, we mentioned these satellites being very important to keep in mind when we talk about the course of infection with HIV. There we go. So as a macrophage does its job, it's really going to chomp up everything in its path and then express it on its surface. So this is one of many antigen-presenting cells, or APCs. So in order for other cells in the immune system to be able to see a virus, particularly, they're going to need a presenting cell, like a macrophage, to present on its surface. And this happens after the macrophage has chomped up everything.
So if you had like a genetic disorder where you didn't have any macrophages in your body, basically you would really be screwed. I mean, like this is the cell that starts everything.
Yeah, absolutely.
So
it is. So this is an innate process. It's not looking for any particular cell to chomp up. But once it does, it'll present these pieces of the virus that might have been in the infected cell on its surface. And it doesn't have to be a specific part of the virus. It's probably presenting things all sorts of ways. One of these pieces, like say this one, is going to be a piece of that HIV virus, and it'll be a protein associated with the virus. So in order to get a specific response to the virus that had infected, whether it's flu like we talked about in the nose or whether it's going to be HIV, it has to be presented. And then a specific response starts with a T helper cell. So Dr. Jacobs described these cells as being the generals of the immune response, or coordinating the immune response that is specific to HIV or any other pathogen.
And in previous semesters, he's also referred to the macrophage as the spy plane. So it'll fly through the battlefield. It'll take pictures of the virus, and then it'll send these pictures to the general, which is the T helper cell. So it might be easier for you to remember if you think of macrophage as a spy plane, which tells the general what they're fighting, and then the general is going to tell the soldiers to fight the good fight. The soldiers, we're going to get into that, but those are the T killer cells. And
yeah, that's a real good point. I think I skipped something. Here we go. Okay, sweet. Yeah, so in order for, that's a really good analogy.
Yeah, I like that one. It stuck with me.
So the specific T helper cell will then send out signals. We can call them, in this case, lymphokines, because the word lymph is going to denote that it's having to do with the immune system. I'm just messing everything up on here. Sorry, Sean. And so we'll use the word lymphokine to be specific to signals sent out by immune cells. So these are different. We're going to use them differently than chemokines, but they are a type of chemokine, or a way that cells are going to communicate between each other. The idea is when it sends out these signals, it's going to tell other cells to be specific. It's actually going to tell others, it's going to make itself divide, basically, so that you have multiple specific T helper cells for the virus. Not multiple, but millions, on the order of millions, based upon a small population subset that you initially had. This way you can have a lot of different cell populations for a lot of different pathogens. As soon as one is presented, a T cell makes contact and starts to divide. Then you have a whole army of generals, basically, with which to coordinate the response.